Hello, this is Professor White, and this is our lab on enzyme. And I will be walking you through with what is going to happen when you perform the lab and some background about enzymes. Now, let's start first with the objectives of our activity for this week. So, first, we are going to identify the reactants and products of a chemical reaction in biology. Also, we're going to distinguish between enzymes, substrates, and reaction products. Additionally, we're going to state the role of enzymes in biological chemical reactions, and as well as list factors that impact or may affect the rate of reaction happening. You're also going to test how conditions such as pH and temperature, and as well as enzyme concentration, may affect the enzyme activity, that case, the enzyme rate of reaction. And lastly, I would like you guys to explain the unit measurement, parts per thousands, as one of the questions in your lab report talks about this one as well. So, without further ado, let's start first with a little background on the importance of chemical reaction in biology. So, chemical reactions are important to biological functions. It basically considers natural functions include when you break down your food, when you're digesting, or in terms of plants, when plants convert light into chemical energy, that is actually a chemical reaction. Additionally, you're also, when you transform anything from reactants to products, that is a chemical reaction. When you bake a cake, there's chemical reaction happening in there. And chemical reaction requires two or more chemicals to interact in a way that modifies the chemical bonds within each of the chemicals and result in a formation of a new substance. So the chemical that interacts as inputs are called reactants, and the ones that is produced are your reaction products. So basically, when we walk through in this lab, you guys will be looking into a chemical reaction. Now, there are some factors that affect chemical reaction. First, the interaction of specific amounts of each of the reactants, or in terms of concentration, for example. Another factor is the presence of, and of course, concentration of an enzyme, which basically can alter the rate of reaction. Same with the pH of the solution, which reaction takes place, and as well as the salinity. So, salinity has to do with dissolved ions of the solution where the reaction is taking place. Last but not least, the temperature of the environment in which the reaction takes place. Now, we are focusing on enzymes. So, what are enzymes? These are basically proteins that folds into very specific shapes in order to interact with various biological molecules. Recall that protein is a biomolecule from our previous lessons, right? So we're going to focus on why are enzymes important? Why are proteins play an important role as a catalyst, as you have to say. Now, enzymes catalyzes biochemical reaction, which means that they assist in ensuring that the reaction will happen and will take place in an energy efficient way. So basically, they are helpers. Catalysts are helpers, but there are parts in your chemical reaction that is involved in it. Enzymes do not add to any products, but instead they are just helpers. So when you say substrates, substrates are reactants. And an example of it would be a sucrose or a disaccharide, carbohydrate. And enzymes, as I've mentioned earlier, they're also called catalysts, and enzymes are proteins. So in this reaction, as you can see on this diagram right here, a substrate entering active site of enzyme. So as you can see, the substrate is the green, and then the active site is the gray. Now, it fits into kind of like a lock and key manner, wherein the active site matches the shape of the substrate. And when they start meeting and getting together, you will see that it kind of fits. Now, the enzyme basically changes the shape slightly as substrates binds. And this is what we call your enzyme substrate complex. Now, as soon as that happens, as you can see over here, the enzyme starts getting involved in the reaction, producing what we call your reaction products. And the product will leave the active site of the enzyme. So that's basically what is happening during the reaction involving enzymes. So, a part of our lab includes solutions and concentrations. So, we'll start first with what solution and concentration. So, living organisms such as bacteria, fungi, squirrels, and even humans are composed of water, and most biological reaction occurs in aqueous solution or water solution of our cell. And as noted, concentration of enzymes in substrates can influence the rate of efficiency of a chemical reaction. So the solution that the solution that's involved are basically consist of two, solvent and solute. Now, the solute represents the substances present in lesser amount dissolved in a solvent, and a solvent, which is present in greater amount, basically is where your solute is dissolved into. So a solvent is water, basically, and then when you mix something in there, that's a solute. Okay, so the ratio of these amounts is directly related to the concentration of a solution. So by definition, the concentration of a solution can be regarded as the amount of solute in a solution. So concentration can be measured in any units, including molality, normality, mass percent, volume percent, or parts per thousand, parts per million, or parts per billion. So we're going to focus on the parts per thousand. So in the lab, you guys will going to convert the percent concentration to parts per thousand. And normally we use this formula: 1% is equal to 10 parts per thousand. Okay, so going to the right side of the slide, as you can see here, the rate of reaction. So reactions are influenced by several factors, and I've mentioned it earlier at the beginning. For example, the enzyme concentration, the substrate concentration, basically affects the amount of reaction products created at a given amount of time. And reaction rate can be understood as a change in the amount over time, and higher rate usually means greater change per unit of time. Let's take a look over here. As I mentioned about, reactions are influenced by several factors. For example, enzyme concentration, substrate concentration, they both affect, right? So the solution with high concentration of substrate will produce more reaction product with enough amount of enzymes, resulting to a higher reaction rate. So you can see here. Okay, so another example here, consider the impact of different enzyme concentrations shown here. Okay, which solution do you think will likely to result in a more reaction product in a given amount of time? Well, the solution with high enzyme concentration will produce more reaction product in a given amount of time than the solution with low enzyme concentration. Now, in the lab, you're going to take a look at what's the effect of the change in temperature and pH to the enzyme reaction rate. So, generally, in terms of temperature, if you increase the temperature, it can lead to either increase in reaction rate or you speed up the reaction. Okay. Now, enzyme are normally very sensitive to high temperature, causing to lose the shape of your enzyme and its functionality. You call this denaturation. Now, in terms of pH, most enzymes function properly only within a very narrow range of pH. So I want you to find that out. What pH is the optimal pH necessary for the enzyme to function? So for the enzymes lab, there'll be three procedures, and you're going to take a look at the following. So the first procedure will be about enzyme reaction rates versus the concentration dependence. This is when you will explore how the enzyme concentration affects the reaction rate. That's your procedure one. Procedure two, the enzyme reaction rate versus temperature dependence. So you're going to explore the how the temperature basically affect enzyme reaction rate. And procedure three is the enzyme reaction rate versus pH dependence. And this part, you're going to explore how the pH affects the enzyme reaction rates. Now, please remember to take a look at the summary of formulas you need for your calculations, because the questions include asking you to solve. And please make sure that you show your solution into your lab report when you answer them. So, first and foremost, you are going to calculate for the reaction rate of the enzyme. So, in this case, you're going to divide the change in the oxygen concentration divided by the time interval. So, the time interval is just always 20 seconds. So, when you divide the parts per thousands of the oxygen concentration change divided by 20 seconds, then you get the reaction rate. Okay, so in this particular example, if the oxygen change is 13.35 parts per thousand divided by 30 seconds, then you get the enzyme reaction rate, which is 0.445 parts per thousand per second. Okay, so that's basically it. Also, in one of the questions, actually two, you are asked to generate a graph for your data. So here's a quick walkthrough, and I'm also going to post a video on how to insert a graph. So step one, you click insert, and then click the chart. And then step three, you're going to click a line graph. It's up to you whether you can be adventurous and check it out, but I'm going to choose line graph here. And then among those line graph options, you choose whichever you like. I actually chose the first one on here for my example. Okay, so right after that, it's going to lead you to automatically add the chart, as you can see here on the left hand side. After you select the line graph, it's going to show you this particular graph inserted right away to your Word document. So all you need to do after step five, an Excel spreadsheet will automatically be generated as soon as you insert the graph on your document. And you are going to manipulate the values on those columns. You're going to change the labels into trials. And again, I will be posting a video on how to make the graph so you can see it up close. Now, if you have any questions, as always, please let me know if you do. And just make sure that you complete your discussion post, and make sure that you also have those sites that you use as your reliable source to back up your response. I'll see you and have a great day.